everyone. Today we'll be solving chapter on chromosome inheritance. This is chapter 19 in human biology, Sylvia S.Mader, the 14th edition. Question number 1. The point of attachment for two sister chromatids is the centromere. Basically, for example, if these are chromosomes, this will be the other. Definitely this is not the shape, but yeah, so the ball or the circle connecting them is the centromere. Each of the following is or are produced by the process of DNA replication. Okay, DNA replication will produce sister chromatids. Chromatin centromere karyotype is produced by the process of DNA replication. So since they gave me a hint is or are, when your DNA replicates, we said that it will form sister chromatids, right? So these sister chromatids, they are connected or attached. Point of attachment is a centromere. Question number 3. Label the drawing of the cell cycle, then list the main events of the stages. So let's start with, so this is the purple arrow, this is all interphase. So the first stage of interphase is the G1, then you have the S phase, then you have the G2, then E, it will be mitosis or cell division, let's just say cell division, and then F is cytokinesis, which is the boring of the cell cytoplasm. At which of the following checkpoints is the DNA checked for damage? And if the damage is present, the cell is placed in the G0 phase. So this is actually during, it will occur during the G1 phase, because just simple rule, it's G1. If there's any damage, if there's anything wrong, it will go to G0. Question 5 to 9 matches statement to the phase of meiosis in the key. So I have the metaphase, prometaphase, telophase, prophase, anaphase. Okay. Chromosomes line up at the equator. So this is the metaphase. Centromeres splits and sister chromatids move to opposite poles. So this is the anaphase. The nucleus dissolves and chromosomes condense. So they're condensed, not they are the condenses, they basically become thicker and visible. So this is during the prophase. The nucleus reforms. It will reform at the end. So during the telophase, spindle fibers attached to the sister chromatids. So this is prometaphase, in which the spindle fibers, they will try to line up the chromosomes in the center of the cell or the equator of the cell. Question number 10. If a parent cell has a diploid number of 18 chromosomes, before meiosis, how many chromosomes will the daughter cells have? So this is meiosis, same numbers, same thing. So if they have diploid number of 18 chromosomes, they're also going to have a diploid number of 18 chromosomes. If a parent cell has 22 chromosomes, the daughter cell following meiosis 2, the second stage of meiosis, will have half haploid. So half 22, it is 11 chromosomes. Crossing over occurs between sister chromatids of the same chromosomes, chromatids of non-homologous pairs. Yes, non-sister chromatids of a homologous pair. Sorry, it's not chromatids of non-homologous pairs. It is non-sister, so not on the same chromosome, not sister chromatids of a homologous pair. Okay, for example, let's say the chromosome for let's say chromosome 17 from your mom is close by during obviously cell division to chromosome 17 from your dad. So what will happen is that one sister chromatids from your mom's homologous pair will exchange some genes with other sister chromatids from your father's gene pair. Question number 13. Which of these helps provide genetic diversity? Independent alignment during metaphase 1, crossing over during prophase 1, and infusion of sperm and egg nuclei during fertilization. All of these are correct. Independent assortment, crossing over, and random. Like you can't decide each time like that. Oh, this sperm with these genes is gonna fertilize with this egg with these genes. No, it's just random. You don't know which of will undergo fertilization with which sperm. Polar bodies are produced during DNA replication, mitosis, spermatogenesis, or genesis. This is basically the process of forming sperm and process of forming ova. So these polar bodies, they will form during the meiosis or the formation of an ovum, and they're basically like small eggs but with the same genes as the ovum, but they have less cytoplasm. And we know that the ovum, it has a large cytoplasm, especially like making its size larger compared with the size of a sperm. Why? Because the cytoplasm, it will contain nutrients to sustain the zygote. Question number 15. The pairing of homologous chromosomes occur during which of the following? So pairing of homologous chromosomes, mitosis, yes. Meiosis 1. Meiosis 1 is basically the homologous pairs, yes. Meiosis 2, no, it's just sister chromatids. Meiosis 2, so I do not have the option for A and B, so there's something wrong. A, mitosis, homologous. When we know homologous chromosomes, we mean homologous pairs. So they occurs to the pairing. So pairing is basically when they're close to each other, and in mitosis, it's just like I like to think about it as, for example, this is the homologous pair 1, this is the other homologous pair. But in mitosis, this is mitosis. But in meiosis, what will happen is, for example, this is a homologous pair 16, and then the other homologous pair is going to be close to it like this. So you see the difference? Here they're like next to each other, and here they are in a line, kinda. So it will only happen in meiosis 1. Question number 16. Sister chromatids separate during anaphase of which of the following? So we are talking about sister chromatids. Mitosis, yes. Meiosis 1, no, it's the homologous pairs that are the ones that will separate, not sister chromatids. Meiosis 2, yes, it involves the separation of sister chromatids, so it is both A and C. Monosomy or trisomy, which is basically having one chromosome or having 3 chromosomes, occurs because of crossing over, inversion, translocation, disjunction. So disjunction is basically what? It is when during meiosis, for example, the homologous pair, or for example, sister chromatids, let's just to make it easier, they do not separate. One of the sperms or one of the ovum, they will have 2 sister chromatids. And so if it further gets into fertilization, the individual, for example, let's take about 6 chromosomes, they might have XXY, XYY. So you understood the idea. Or they might, and if one of the daughter cells takes 2 chromosomes, the other daughter cell, what is gonna have? It's gonna have 0. So this is true in Turner syndrome, in which you only have X the other X is missing. Okay, so this is basically the process of non-disjunction. Question number 18. A person with Klinefelter syndrome is what? So this is a name of a syndrome, and it affects males because there's a Y chromosomes. So they are males. And what do they have? I think they have XXY. YY. YY. No, I don't think they have YY. I think they have XXY. This was the last question. Anyway, thanks for watching and hope you benefited from the video. And if you have any questions, drop it in the comments.